This is lesson 3, chapter cell cycle and cell division. Students, in this video, I will discuss about the second type of cell division. This is called as meiosis cell division, and it is also called as reductional cell division. Now, what is meiosis? Meiosis, it is a type of cell division in which the germ mother cells of sexually reproducing plants and animals divides to form gametes in which the chromosome number is reduced to half. And therefore, the meiosis cell division, it is also called as reductional cell division. So let me repeat a few things from the last video now. So as you know that during the mitotic cell division, so there the mother cell, it always gave rise to two daughter cells, and these two daughter cells, they had the same components as that of the mother cell. As for example, if the mother cell have 10 chromosomes, the daughter cells will also possess 10 chromosomes each. Now, in case of meiosis cell division, so this is somewhat different. Meiosis, here, this is a cell division which takes place in the reproductive cells or in the reproductive organs of the living organisms. And here specifically it is said that sexually reproducing plants and animals. So here what happens that the mother cell, which here in the definition it is called to be germ mother cell. So mother cell, it divides and it forms 4 daughter cells, and these 4 daughter cells possess half the number of chromosome from the mother cell. Say for example, if the mother cell again it possesses 10 chromosomes, here the daughter cells, it will have only 5 chromosomes each. That means from the mother cell the number of chromosome had reduced to half. And in that way, the resultant cells, those daughter cells we call them to be gametes, which are formed during sexual reproduction process, either in the plant world, animal world, or any kind of living organism which undergoes sexual reproduction. I hope now you have understood why this meiosis cell division, it is also called as reductional cell division process. Here, because the number of chromosome number or the total content of the DNA, that gets reduced to half, and that is distributed to the daughter cells equally, and therefore the daughter cells, they possess half the number of chromosome, and in that way we find the gametes, they have the haploid set of chromosome number. Now if we compare it between the mitotic and the meiotic cell division, mitotic cells, they always possess diploid, that is twice n is equal to 10, as I have given the example of 10 chromosomes in the specific cell. So there that particular condition, it is called as diploid, it is twice n. And in case of meiosis cell division or meiotic cell division, which specifically takes place in sexually reproducing organism in the reproductive cells, here it is taking place in the reproductive cells. So the resultant cells, they will be called as haploid because there the number of chromosome, that is n, is equal to half. That means here it will be 5 from the previously given example. So I hope this particular point, it is clear to all of you. Now let's come to the stages or different steps that takes place during meiosis cell division. So in case of meiosis cell division, so here also the karyokinesis and cytokinesis takes place, and the meiosis cell division, here it is mainly divided into two stages. One is meiosis 1 and another one is meiosis 2. In meiosis 1, also karyokinesis and cytokinesis will take place. In meiosis 2, also karyokinesis and cytokinesis will take place. Now let's discuss about what happens in meiosis 1 and what happens in meiosis 2, that how we can see the division of chromosome that gets reduced to half. Students, so let's discuss about the meiosis cell division, that is meiosis 1. So this meiosis 1, it is divided into firstly prophase 1, then metaphase 1, then thirdly anaphase 1, fourth is telophase 1, and the fifth step that it takes place here, that is cytokinesis 1. So these are the 4 stages that happens during meiosis 1 cell division. So this is meiosis as a whole, and as I have said that meiosis, it takes place in 2 steps. One is meiosis 1 and meiosis 2. So under meiosis 1, so these are the phases. Again, in prophase 1 that is divided into 5 phases. And those phases are leptotin, zygotin, pectitin, diplotin, and dikinesis. So in total, in meiosis 1, there are 5, 6, 7, 8, 9. There are 9 stages. So that we have to study first. And in meiosis 2, so meiosis 2 again, so that is a different phase. So let's discuss about the meiosis 1 division first. Now let's discuss about meiosis, the different stages in meiosis 1. And here at first it comes prophase. So in prophase generally, so this is the cell, and here this is the nucleus. So this nucleus, it has long threads of chromosomes. So here the chromosomes, they are thin, slender, thread-like structures. And in prophase, we find the chromosomes with 2 chromatids. So these are the 2 chromatids having single arm. So after the cell cycle, so here the chromosomes, they have become condensed, so, or they are in the process of condensation. Therefore, we find the chromosomes during prophase, and this is the first stage of prophase 1, which is leptotin. So as I said that the prophase 1, it is divided into 5 stages again. So this is the leptotin, and here it have the nuclear membrane along with the nucleolus. So it is present there. So after this comes the next stage. So this is called as zygotin. So here I have considered a total of 4 chromosomes. That is, here the total number of chromosomes, it is in diploid form. Twice n is equal to 4. So here this is the parent cell with 4 chromosomes. Now what will happen in the zygotin stage? So leptotin is followed by zygotin. So here again the nucleus. So this is the nucleus. So this is the nucleus. So this is the nucleus again. So here the chromosomes, they becomes 2-armed. The chromatids, in the chromatids, at first the arms, there were only 1. So here the chromatids, they get extended into 2 arms, and in this condition we call this to be bivalent condition. So here we can see the bivalent condition of the chromosomes. So here there were 4 chromosomes. So here I have drawn the 4 chromosomes. And here one more specific character takes place in zygote aru hetu hoise that the homologous chromosome they get pairs up. So here we find homologous pair of chromosomes as the like chromosomes. See, within a cell or within the nucleus we bear two types of chromosome, and that is based on the transmission of characters from one generation to the other. Half of the chromosome it comes from the father, half of the chromosome it comes from the mother, and therefore we have means like pair of chromosome, and those like pair of chromosome which possesses the same genes, that is only called as homologous pair. So in zygote, this specific character that gets completed, here the homologous chromosome it gets paired or they come closer to each other, and there, say for example, these two are the homologous chromosome now. Is that okay? So these two are the homologous chromosome where they have the like genes, and here one more event takes place. So this is synapsis. Synapsis, that means the homologous pair they comes closer to each other, and that particular phenomenon that is only called as synapsis. So these three events it takes place in zygote of prophase 1. So as I am discussing about prophase 1 now, so these are the two steps. Now zygote it is followed by Pectide. So this is the cell again, and this is the nuclear membrane. Now nucleolus it is already present there. Now in this case the nucleolus it slowly starts disappearing, and the nuclear membrane, so this is the nuclear membrane. So nuclear membrane this also starts to dissolve. The nuclear membrane also starts to dissolve. Now what will happen inside the nucleus? So as you have seen here that the synapsis takes place during zygote. Now the synapsis that gets more deeper, and here we find a very significant phase that takes place during meiosis cell division, and this phase this is called as crossing over. So the synapsis it gets deeper, and because of that homologous chromosome it undergoes exchange of the chromatids. That means the non-sister chromatid of the homologous pair of chromosome they exchange the chromatid arm, and this exchange this is only called as crossing over that takes place during the Pectide of prophase 1. So this is a very important step that takes place in every sexually reproducing organism, that here the chromosomal exchange between non-sister chromatid. Non-sister chromatid, see here, say for example this is the chromosome. So these are the sister chromatids in a chromosome, and this is the other chromosome in this homologous pair. So these two chromosomes they have the same type of genes which expresses a particular character. Now what happens that this chromatid and this chromatid they becomes the non-sister chromatid of this homologous pair. So during crossing over they exchange their chromatid arms, and that is only we call it as crossing over, and this happens in the Pectide stage of prophase 1. So here there were, as you know, that there were 4 chromosomes. So here crossing over have taken place. So this we can show it in this way. Now next to Pectide comes the next stage that is called as diplotin. So in diplotin what happens here? Here in diplotin the crossing over chromosome. So here the nuclear membrane, it starts disappearing. So here I have drawn the dotted line, and the nucleolus, it also starts disappearing. So in this now we find chiasmata. Now what is chiasmata? These are some points of attachment. Say for example, this is the point that is attached now after the process of crossing over, and this is the point where it is attached now. So the point of attachment, so in the crossover chromosome, that is called as chiasmata, because the crossing over directly, it do not lead to the separation of the chromatids. So they start separating slowly, and there are still some points where the chromosomes are attached to each other. That means the non-sister chromatic between the homologous pair of chromosome, that remains attached, and that point of attachment is only called as chiasmata. So in diplotin, this chiasmata, it is found, and it starts to reappear as a normal chromosome again. So here these are the point of attachment that we find in diplotin stage, and these are also regarded as dyads. Why these are regarded as dyads? Because now they appears to be, now this chromosomes, they appears to be in a F-shaped manner. So this point of attachment, so this is the point of attachment here, and this is one chromosome, this is one chromosome of two different chromosomes. Though they are homologous chromosome, but still they are two different chromosomes. So here, as this non-sister chromatid, they are attached at that particular point. So here what we find an F-shaped pattern. So that is why this particular condition, this is called as the dyad condition. So that also we can see in diplotin of prophase 1, and followed by diplotin is the last of prophase 1, last stage of prophase 1. So this is called as dikinesis. So here the nuclear membrane, it becomes disappeared, it gets disorganized, and now what happens, that the chiasma, it reaches the terminal end. Now the chiasma, it reaches the terminal end of the two homologous, or of the two chromosomes of the homologous pair, and this we term it as terminalization of chiasma. So this we term it as terminalization of chiasma, because here the chiasma is directly do not separate from the chromosome. So it slowly goes towards the terminal position of the chromatid. So therefore this is called as terminalization of chiasma, and till the end of dikinesis, so they become separated, or they again reappears to be like the normal chromosome after the process of crossing over. So let me draw here all the 4 chromosomes. So these are the 4 chromosomes that I considered at the very first of this cell division process. So here the 4 chromosomes, they are still there. Is that okay? And this is the last stage of dikinesis, where the significant part, that is the terminalization of chiasma, and later was the latter part of dikinesis. What happens, that terminalization of chiasma, that gets separated. And another difference what we can see here is that the nuclear membrane, right from the stage of lactotin, it starts to disappear. So in lactotin, there the nuclear membrane, it was intact. In digotin, it was there, but from the latter part of pectin, then from diplotin and dikinases, the nuclear membrane and the nucleolus, that slowly starts disappearing. So this is all about the prophase 1. Now after prophase 1 comes metaphase 1. Now let's see what happens in metaphase 1. So in metaphase 1, this is the cell. As you know that the nuclear membrane, it completely disappears till metaphase 1 of meiosis 1. So here the chromosomes, they comes and lie in the equatorial plane. All the 4 chromosomes from the parent cell, they lie and they come and lie there in the equatorial plane. And here the spindle fibers also gets formed. The spindle fibers, they starts appearing. Actually it starts appearing from the prophase only, but as we have that I have already discussed in during mitosis, so I have not mentioned about that there. So here I will draw the spindle fibers as well. And as you know that the spindle fibers in case of an animal cell, so in case of an animal cell, so there it will be formed from the centriole. But in case of plant cells, so we find only the astral rays which are formed from the microtubules. So that will be same. Just only the difference will come only with the distribution of the chromosome. So in metaphase now, so this is metaphase. So in metaphase, the chromosomes, they will come and they will lie in the equatorial plane. And there is no nuclear membrane or the nucleolus. The nuclear membrane and the nucleolus, they will completely disappear. So this happens in metaphase 1. So from metaphase 1, that is followed, the metaphase 1 gets followed by anaphase 1. Now what happens in anaphase 1? Now see, these were the homologous chromosome. These two were the homologous chromosome. Now in meiosis 1, in case of anaphase, so in mitosis also you have got these particular stages. Now what will happen here? Here in anaphase, the chromosomes, they will be pulled again to the polar ends of the cell. Here they will be pulled towards the polar end of the cell. But what will happen here, that the homologous chromosomes, they will get separated, and they will, one from each of the homologous chromosome, they will go to different poles. Yat ki hobo, jikhini homologous chromosome ase, dutai edokor pora eta metaphase pora eta tahante ki hoy jabo, they will get separated, and both the homologous chromosome, they will go to 2 different poles. So here, say for example, this were the homologous chromosome. Now these are the 2. Now they will get separated. Say for example, this homologous chromosome, they will travel towards one pole, and the other homologous chromosome, they will go towards the other pole. Similarly, here also, eta ki hobo, dutar bhitorot eta polole jabo, and the other will go to the other pole. Is it okay? The spindle fibers, they will separate the homologous chromosome. In very simple words, the spindle fibers, they will separate the homologous chromosome, and separate the homologous chromosome, and it will reach the 2 different poles there in the cell. So this happens in anaphase of meiosis 1. Is that okay? So this is again a very important step that takes place during meiosis cell division. Now from this 4, now 2 it went upward and 2 it went towards the lower pole. Now what will happen? After anaphase comes the process of telophase. So after the chromosomes, now from the 4, 2 chromosomes that is present in each of the 2 poles. Now what will happen? During telophase 1, During telophase 1, here the nuclear membrane, it will again reappear, and now this parent cell, now it have how many nucleus? It has 2 nucleus. Is that okay? And these 2 nucleus from the parent cell, which possesses 4 chromosomes, now this 2 nucleus, it contains half the number of chromosomes from the parent cell. Is that okay? So as you know that during anaphase, the homologous pair, they got separated, they got segregated and reached 2 different poles. So here, now in this parent cell, there will be 2 nucleus, but here the nucleus, it will bear half the number of chromosomes, and therefore here the number of chromosomes, it got reduced, and just after this will start the process of cytokinesis 1. So here the cell plate will form here, the cell plate it will form here, and now this 2 nucleus, it possesses only 2 chromosomes. Is that okay? And finally it will give rise to 2 daughter cell. So this is the 2 daughter cell, and the chromosomal content, it again gets disorganized. Is that okay? So now in both these two cells, the number of chromosome, it will be n. And how we are going to write this? As we are going to write it as n is equal to 2, n is equal to 2. Here that means what? The number of chromosome is 2 here. But in the parent cell, so there the number of chromosome, it was twice, and it is equal to 4. But till the last part of this meiosis 1, so which finished in cytokinesis 1, so there were 2 nucleus, and those 2 nucleus, it bear half the number of chromosomes than the mother cell, or the cell that was first grown in lactotin stage. Is it okay? So as it is a continuous process, so at last in cytokinesis 1, we have 2 cells with 2 nucleus, and this 2 nucleus, it possesses haploid set of number, that means half the number of chromosomes. So as this is a continuous process, now what will happen here is that meiosis 2 will start. After meiosis 1, till cytokinesis 1, it was all part of meiosis 1. Now here the second phase of cell division, it will start with the 2 daughter cells. Is that okay? And here the number of chromosome, it is 2 from the parent cell. Here the number of chromosome, it got reduced to half, and it has the nucleus. So again it will start with the interphase, as this is a part of the cell cycle. So yaar ki hoba aako interphase opor aako start hobo, and interphase opor start hobo pisot ki hobo. So this is now in interphase stage. Again the meiosis 2 will start with prophase 1. So prophase 1 ki hobo, etu duta cell ke homane hoi thakibo. Prophase 1 ki hobo, again it will form the chromosome, but here only 2 chromosomes will be there in the parent cells. Is it okay? After cytokinesis 1, so these are the 2 cells. Is that okay? And here the number of chromosome, it was 2. So slowly, so this is prophase. So this is prophase 2. Prophase 2. After prophase 2, so this is same as that of mitosis. Only the first part that was different. So this is same as that of mitosis. So here the nuclear membrane, towards the latter part of prophase 2, the nuclear membrane, it will disappear. Is that okay? And that will mark the start of metaphase 2. So in metaphase 2, what will happen? So these are the 2 chromosomes from the daughter cells of the parent cell. Is it okay? So these are the 2 chromosomes each in the 2 daughter cells. So here spindle fibers will be formed here. Is it okay? So spindle fibers, they are here. Is it okay? So here what will happen again? So metaphase 2 is followed by anaphase 2. So as you know, this will be meiosis 2, it will be same as that of mitosis. So here what will happen? The spindle fibers, it will separate. It will come and attach to the centromere and it will separate the centromere. And from each of the chromosome, now as the chromatids, they are attached to the centromere, and here in anaphase the spindle fibers, they get attached to the centromere, and what will this spindle fibers do? They will pull the chromatids towards both the poles. In other case, during anaphase 2, the homologous chromosomes, they went to different poles, but here from the same chromosome, half of the chromatid it will go to one pole and the other half that will go to the other pole. So that will be anaphase 1. Is it okay? So anaphase 1, so finally we will find the chromatids in the poles. Is it okay? So these are the spindle fibers here. So it will reach the poles. So here again this got separated in both the cells. Let me draw it properly here. So these are the 2 chromosomes that reach the pole. Is it okay? So these are the 2 daughter cells. So these are 2 daughter cells again, and this is again followed by telophase 2. So in telophase 2 again, these chromosomes, they will again get surrounded by the nuclear membrane. Is that okay? So these are the 2 chromosomes here. So now in each of the cells, how many nucleus will be there? 2 nucleus will be there in each of the cells. And after telophase, what is the last step? So that is cytokinesis 2. So in cytokinesis 2, what will happen? That a cell plate will form in between these 2 cells, and from this, how many resultant cells that we get? We get 4 resultant cells with 2 chromosomes each. Is that okay? The resultant cell, it will be 4. Is that okay? So this 2 cell, or all these 2 cells, they were formed from meiosis 1. Is that okay? Try to remember that this is a continuous process. So we got these 2 cells from meiosis 1, as there one cytokinesis has happened. So now the cell division, it will again start in these 2 daughter cells, and it will be just same as that of mitosis. So after mitotic cell division, which we regarded as meiosis 2, is that okay? So we find 4 daughter cells, and therefore in the definition it was given that meiosis, it gives rise to 4 daughter cells, and there the number of chromosome, it will reduce to half from the parent cell. So as in case of this, what was the actual chromosome number or diploid chromosome number? That was 4. But till the last step, so how many chromosome numbers that we find here? So it is 2. Each of the daughter cells, they possesses only 2 chromosomes from the parent cell. Is it okay? And in all this process, or in all the steps, there the main events that took place, it was the division of the nucleus and the division of the cytoplasm. At first, division of nucleus will take place, and then the division of cytoplasm will take place. Is that okay? So this is all about meiosis cell division.